Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes we have talked about the transcriptome assembly process, then we have talked about the transcriptome quantification process, and in between we had a very short hands on how to get to the transcript abundance part or the raw count part from the read data. So, we will talk about the biases in RNA sequencing experiments in this class. So, we have touched upon this topic very briefly in the last class when we were talking about the alignment free methods, and we will talk about these biases in a lot more detail in this class. Now, this is very important because these biases can affect our quality of analysis, our quality of inferences that we draw from these RNA sequencing experiments, right. So, we need to understand these different types of biases that are present there. Sometimes we may not get to the exact origin of these biases, right, why these biases arise, right. So, this is something we still cannot predict, but we observe these biases in the data, and this is something that we need to correct for before we can proceed for further downstream analysis. So, these are the concepts that we will be discussing in today's class. So, the biases in RNA sequencing experiments and methods for bias correction. Okay. So, let us look into these different types of biases, and here are the keywords. So, we will talk about positional bias, sequence specific bias, and finally, we will introduce the term normalization. Okay. So, let us start with a very simple example of this RNA sequencing experiment, right. So, at the end of an RNA sequencing experiment, the goal is to measure or quantify expression of genes in a sample, right, and maybe compare these expressions across different samples, right. So, maybe different conditions, different treatments, etc., and see which genes are showing higher expression, which genes are showing lower expression. This gives us a tremendous insight into the what is actually happening inside the body or inside the cell, etc. So, let us look at this example, right. So, we have these three genes, right, gene A, gene B, gene C, and we have these blue reads mapping to these genes. Okay. So, we are not talking about exons or isoforms, just keep it very simple here. So, we are seeing these reads that map to gene A, gene B and gene C in an RNA-seq experiment. Okay. And what we get is we generate a raw count data, right. This is something we have seen, right. We have this for many genes, we have these raw counts, right, 4, 5 or thousands, right. So, here is an example from, and when you convert this to numbers, right, number of reads mapped to each gene, we see for gene A we have 6 reads mapping to this gene, right. Then for gene B we have 3 reads, and for gene C we have 2 reads, right. So, this is something what we see. Now, the goal, as I said, is to go to the expression level of genes. Okay. So, by looking at this table, can we just say gene A has higher expression than gene B, right. So, can we just simply say because gene A shows higher number of reads, right, mapping to it, right. So, we will talk about this, right. This is not so simple, and we will talk about, right, how we can actually arrive at the expression level from this raw read count data. Okay. So, we can actually extend this example further and imagine we have now 3 samples, sample 1, sample 2 and sample 3, okay. And we do the mapping as before, and we see these counts, right, number of reads that map to each of these genes in each of these samples, right. So, we have sample 1, 2, 3 and we have gene ABC. Okay. For sample 1 we see 6, 3, 2, right, reads mapping to gene ABC respectively. Then we have for sample 2 we have these numbers 12, 6 and 4, and for sample 3 we have 0, 7 and 4. Okay. Now, the question is does this mean gene A has higher expression in sample 2 compared to sample 1, because you see in sample 2 there are 12 reads that are mapping to gene A, whereas in sample 1 there are 6 reads mapping to gene A. Okay. Does this mean this gene A is highly expressed in this sample 2? Okay. This is a question again we need to answer, because we do a lot of this kind of analysis, right, where we compare this expression of the same gene across different samples, different conditions, maybe different treatments. The second question is, right, if you look at this data again, right. So, for gene B and C does this mean sample 3 has higher expression of gene B and C compared to sample 1, right. So, you see again the number of reads that are mapping to gene B in sample 3 is 7, right, whereas this is 3 in sample 1, and similarly for gene C we have 4 reads mapping to sample gene C in sample 3, and we have 2 reads in sample 1. So, the question is that so you can say B and C are showing higher expression in sample 3, is that so? Again the answers are not so simple, because there are a lot of other factors that we need to take into account, okay, and this is something that we will talk about in this class. Okay. So, whenever you do an RNA-seq experiment, you will always get something called technical variation, and then we have biological variation, okay. And the goal of this discussion on biases and bias correction is to actually minimize this technical variation, so that we can study this biological variation, right, because that is where the insight is, right. So, we understand the behavior of the system, we understand how the cell is responding to certain treatment, right, or maybe responding to certain environment, etc. Okay, so, let us now move into the biases in RNA-seq data, right. So, what are the different types of biases that we can see, okay, and these biases arise due to nature of transcripts during the library preparation steps and the sequencing steps. Okay, so, in the next slides we will talk about like these steps where these biases come into picture. Okay. So, the first step in RNA sequencing experiment, if you remember the flow chart, is the sample preparation, right. So, we have sample storage sometimes, right. If you are working with tissue data, you have to collect these tissues and maybe you have to store them in certain conditions, right. So, and then you have the RNA preparation part, right. So, if you are isolating RNA, right, you will follow certain protocols that we can affect these RNA-seq data quality, right, and which kind of what kind of reads data you will get. Similarly, we have the library preparation step that can also introduce some biases in the data. You have sequencing step, again depending on the sequencing platform, you have amplification in some platforms, etc. Those can affect the quality or maybe introduce some biases in the data, and of course, there are some inherent biases of RNA-seq experiments, which we will talk about at the end of this class. Okay, so, let us now take each of these points and see how they actually introduce biases in the data. Okay, so, the first step is the sample storage and RNA preparation, as we have mentioned. So, we have different types of RNA isolation protocols, and this might affect the RNA-seq data, right. So, you can have different types of biases, different types of RNA processes that you are utilizing that actually will determine, right. So, again the quality of prepared RNA is very important here, as you can probably understand, right, because there is a lot of these RNase molecules around. So, you can get maybe partial degradation of some molecules, and maybe some RNA molecules are degraded, and this can affect or this can bias your data, right. So, you can imagine, right, so if out of the full transcript, if some of the transcripts are degraded by these RNAs, right, you are biasing your data. You will see maybe some genes are lowly expressed and maybe in actual scenario these transcripts have been degraded by presence of RNAs, right. So, this is very important, right. The quality of prepared RNA is a very important step, and as I mentioned, you can see this partial RNA degradation, and if you have this partial RNA degradation, that will bias your data, right. So, again this is an experimental step that can ultimately affect the data that you see after next generation sequencing. Okay. So, is there any way we can actually deal with this, right. So, this is something that is very important, and we will come to that towards the end of this class. Now, moving on to the library preparation steps, right. So, there are different steps when you prepare an RNA-seq library, we have talked about them, right. So, one is fragmentation, right. So, we have to fragment long transcripts and at different steps, and you have the reverse transcription, right. We have talked about this. So, this is generation of cDNA, double stranded cDNA, and then sometimes you also have PCR amplification, right. If you are going into Illumina or ion torrent platforms, you need to have on bead amplification or cluster generation, right. So, bridge amplification. So, that can also introduce bias in the RNA-seq data. So, let us talk about the fragmentation, how it can actually affect the RNA-seq data. So, we usually have random fragmentation, right. So, this can happen at any place in the transcript, and you can do the fragmentation either in the RNA level, right. You can fragment the RNA, or you can do the fragmentation at cDNA, right. And what happens is that after this fragmentation, because this is a random process, you generate different size, right, fragments of different size, and before you go for sequencing, you have to do something called a size selection. Okay. Again, if these fragmentation processes are biased towards certain sites, right, you can see only certain fragments are getting selected at the size selection step, and long fragments are excluded from sequencing. Okay. And this has been observed, right, in many experiments, that these starting points of fragments, they actually coincide quite often. Okay. This is not supposed to be the case, right. So, if fragmentation is random, then you should see a lot of these different starting points for fragments, right. But what researchers have observed, that these fragments, their starting points often coincide, suggesting that this is not a random process, right. So, we think this is a random fragmentation process, this is not. Okay. And again, this introduces bias in the data, which we need to account for. Okay. So, we have to do something about this at the bioinformatics step, right. We cannot probably change these protocols too much, maybe there is some sort of improvements that we can make, but then we have to have these bias correction steps in the bioinformatic pipeline. Then we have the reverse transcription, right. So, for many library preparation steps, we need to do the reverse transcription, except probably the direct RNA sequencing that we have talked about. So, in reverse transcription, the earlier researchers used this oligo-DT primer, where they are looking at the poly A tail, right. So, these primers will bind at the poly A tail. So, what you will see when you have this kind of oligo-DT primer you are using, you will see like only certain parts, right, from the 3' end of the RNA, they will be amplified, and maybe the you will see a lot of over representation of the 3' end of the RNA molecules, right. So, you will see more reads there, and you will see less reads from the 5' end. Okay. So, this can happen, and again we have talked about this. This means we have non-uniform coverage across the transcript. Now, what researchers suggested, maybe we can use random primers instead of this oligo-DT, right. So, this should solve the purpose, solve this problem of this bias towards certain region, right. But again, we have binding preferences, right. These are usually random primers. They will again have binding preferences for certain regions, or which we call as priming bias, and this can again lead to generation of only certain fragments being overrepresented in the read data. Okay. So, again you can try different ways, but again these biases will be there in the data. You can also have bias due to amplification step, right. So, this is amplification in the cDNA synthesis step or in the cluster generation on on-beat amplification, right. So, you can generate this bias. Now, we have talked about priming bias. So, means when we are getting only certain fragments that will be amplified. So, these will be overrepresented in the library. In addition, there is something called a GC content bias. Okay. So, what is this GC content bias? Is that what we have observed, or researchers have observed, is that sequences that have low or high GC, these are usually poorly amplified, and they are poorly represented in the RNA-seq library. Okay. So, again this is something to do with the amplification process, and you can have certain modifications in the amplification step, right. This is an experimental modification. You can add certain solutions that will help in better amplification of these low or high GC regions, right. So, that you get more or less uniform representation of all these regions in the RNA-seq library. So, you have some experimental solutions, but again you do not know how effective this solution is until you see the read data, and in the from the read data you can identify whether this kind of biases are still there. Okay. And this is something that is a very important part of all this analysis. And finally, what we have talked about is that there are inherent biases in the data, right. So, one of the first thing is the variation in gene length. Okay. So, you have let us say 10,000 genes, right. They vary tremendously in terms of the size, right, of the transcript. So, some transcripts may be in 1 kb length, some transcripts may be only 200 base pairs, some transcripts may be 5 kb, right, and this also leads to bias in the data. So, we will talk about this, right. So, what happens is that if you have a longer transcript, right, there is a high chance that you will get more fragments, right, and you will have more reads coming from this transcript in the RNA-seq library, right, because simply because this is longer transcript, you have more fragments generated and that will be sequenced more than a transcript that is smaller in length. Okay. So, this is what actually would lead to bias in the data. Okay. So, you will find longer genes will have more reads mapping to them usually. If you are comparing this between a long gene and a short gene, right, and they are expressed at same level, you will usually see longer genes will have more reach compared to the short gene. Okay. So, this is a bias that is there in the data. In addition, you have something called variation in sequencing depth from one sample to another, right. So, if you sequence two samples, maybe in one sample you get 1 million reads in total, in another sample you will probably get maybe 1.5 million reads, right. So, again you cannot control how many reads exactly you will get from each sample, right. So, you will see this variation, and again you need to account for this variation. Okay. So, we have talked about these different types of biases in the data that you can see. Some are sample specific biases, right. So, when I say sample specific biases mean these are present in each sample and these are unique, right. So, they are unique for sample, right. This is not like you have processing these 8 samples and you see this same bias across this sample. That will actually be easier for us to deal with, because if you see this kind of uniform biases across this data set, you can filter out these biases quite easily, or maybe if you are doing comparative analysis between samples, you may not have to counter or account for these kind of biases. But what we see is that these biases are sample specific, okay, and this means we have to correct for these biases at the sample level. Okay. And this, as I mentioned, this is unique to each sample. So, they vary from one sample to another. You have sequence specific bias, this is due to priming bias, right. So, again if you are using random primers for example reverse transcription, you will see only certain regions will bind these primers, random primers, they are amplified much more efficiently, right. Again this is something we cannot really do anything about because if you are using these random primers, and this will lead to something called sequence specific bias, right. So, again these regions that bind to these primers will be overrepresented in the RNA-seq library, and the other regions will be poorly represented. We have also talked about something called positional bias, right. So, this is again something to do with mRNA processing and degradation, right. So, if you are processing this mRNA, if you are using this oligo DT and maybe some ends, right, let us say one end of the RNA is degraded, right. So, most of the reads will come from a specific position within the transcript. Okay. And then we also talked about this GC content bias, right. So, this arises due to amplification step. So, some regions which neutral GC close to 50%, they are actually amplified much more efficiently than the regions that have very low or very high GC content. And we talked about again some of the experimental improvements that you can make to actually reduce this GC content bias. And then finally, we also have this variation in sequence depth. This again varies from one sample to another. So, one thing you should note, right, we have not included gene length bias here, because this will not vary from one sample to another. This is same across all samples, or likely to be same. Now, the question is, how do we address these biases, right. So, we have understood these different types of sample specific biases, right, and we need to remove these biases from the data before we actually further process the data, right. So, these are technical variations, these are introduced because of the experimental process, the RNA preparation process or the library preparation process, right. These are not actual biological variation. Okay. So, we want to minimize or remove these technical variation, so that we can study the biological variation in the data. Now, how do we address this? So, we can learn about these biases from technical replicates of the same sample. So, we use two terms, one is technical replicates, another is biological replicates, right. So, technical replicates is you are doing the same experiment, right, with the same sample multiple times, okay, and this will help us understand, right. If you see this variation in read data across these technical replicates, you can then say, okay, this is where these are the different types of biases that we see, and we can model these kind of biases. Okay. And we can take different approaches for bias correction, and this has been proposed, right. Many methods have been proposed, and we can discuss some of these methods in the next few slides. Okay. So, one of the methods that have been proposed is it relies on something called generalized additive model. This is the paper, again the reference is given at the end, and it actually does a correction for gene length, GC content and dinucleotide frequencies. So, we have talked about these biases because of gene length, right. So, this again varies from one gene to another. So, this has to be taken into account. We have GC content bias, we again discussed, this arises because of the amplification bias with specific GC contents. Then you have dinucleotide frequencies, again this will arise if you have sequence specific biases, right. So, certain sequences are preferred by the primers that are binding, right. So, you can then see these dinucleotide frequencies changing, right. Some dinucleotide will be much more abundant than the others. Okay. So, this method actually can take into account all these biases and correct for those. You have also something called likelihood based approach for bias correction. Again, we are not going into the details of all these methods. Okay. So, what it does, it is actually looking at this expression variation as well as the bias, right. So, it kind of develops models, right, that actually kind of generate estimates for this bias. Okay. And it can correct for sequence specific and positional bias. There are other methods, for example, this is a method that has been proposed using Bayesian networks based approach. Again, we are not going into the statistics. If you are interested, you can have a look. And you can see this package that has been developed. So, you can take this tool, right, in R and you can actually estimate these biases in your own data set. Okay. So, again we will introduce R when you go into the hands on, and we will see how we can utilize these different packages for application to our own data sets. Okay. So, this is a method that actually can learn the type of bias that is present in the data and can correct for those biases. Now, there are specific methods that have been proposed, for example, for GC content normalization, right. So, we have learned about this GC content bias, right. This arises because of this difference in amplification efficiency between different fragments with different GC content. So, this actually has been proposed by in this paper that is mentioned here, and they have suggested 3 strategies for normalization. So, again you can have a look into the details. We are not going to discuss all these strategies, the different types of methods, etc. And again there is an R package that they have developed, is called EDS-seq, and again here is the link, you can see and download and install if you have some knowledge of R. Otherwise, do not worry, we will actually discuss how actually we can install these R packages and how we actually use them to correct for these biases in our own data sets. So, we will do that in the hands on part. There is another method which is actually relies on a bias modeling framework, and it utilizes something called generalized linear model. Again, these technical terms are not so important. If you are interested in the actual statistics and the methods, please have a look at the original paper. We are not going to go into the details of this. This method can account for positional bias, sequence bias and GC content, right. So, these are the 3 major biases that we have seen, that we have discussed in previously, and they have also developed a package called Alpine, and again it is an R package that you can download and apply in your own data. Now, we will see, right, most of these packages for transcriptomic analysis, these are actually developed as R packages, and we would have to actually use R to analyze these data sets. So, we will do that in the hands on part. So, one of the thing that I have mentioned, right, one of the points I mentioned is that there is a gene length bias, right, and what we originally assume is that the expression level of a gene, right, is proportional to the number of reads that map to the map to the gene, right. So, this is a very naive assumption, right, and again this will be affected by the this gene length bias. Okay. So, this is the assumption, right. So, the number of reads mapped to a gene is proportional to the expression level, but what we see this is not the case, and this actually this number also depends on gene length. Okay. And as we have discussed, right, and you can imagine, right, if you have a longer transcript, you have more fragments generated which can be sequenced in the library. So, these fragments will be overrepresented in the library and they will appear as more highly expressed, right, even though they are not. Okay. So, longer genes, if you compare a long gene and a short gene and compare if they are expressed at the same level, even then you will see the longer gene will show higher number of reads mapped to them, because they simply have more number of fragments generated from them. So, coming back to the same example that I gave you, right. So, again this we have these 3 genes, gene A, gene B, gene C, and we have the count table. And now what we will do, since we know about this gene length bias, I will also mention the gene lengths right here. Okay. So, here you see now gene A is of length 2 kb, right, gene B is 1 kb and gene C is 0.5 kb. Now, once we know this, right, and we know about gene length bias, so can we answer this question now? Okay. Does gene A have higher expression compared to gene B? And the answer is no, right. So, because gene A is longer, so it is actually twice, right. So, the length is 2 kb compared to 1 kb of B, right, and it should have around 2 times more reads mapped to it. Okay. So, this is something again that now tells us, okay, this is not true, right. So, gene A and gene B probably have very similar expression level. Okay. So, you can now see that when we are accounting for these biases, these questions, the answers to the questions now become clear, and we are actually getting the right answers. Okay. We are not making wrong inferences. And we can actually extend this to this other example that we have taken. We are comparing these expression levels across samples, right, sample 1, sample 2, sample 3, and again we have this question, does gene A have higher expression level in sample 2 compared to sample 1, and do genes B and C show lower expression in sample 3 compared to sample 1. Now, what you have done compared to the earlier table is I have now added this total number of reads here. Okay. So, this is the total sequencing depth, right, and as we have discussed, this depth can vary from one sample to another, right. So, this is something we again have to take into account, right, and you can imagine probably, right, if we have total number of reads more in a sample, right, you are likely to see more reads mapping to each gene in that sample, right. So, here if you take this example here, if you have 22 reads in this sample 2 compared to 11 reads in sample 1 and 11 reads in sample 3, right. So, naturally you will expect more reads mapping to gene A, B, C, right, in this sample 2 compared to sample 1. So, what we usually can do is we can actually account for this total number of reads, and we can look at fraction of reads that are mapping to the genes, okay, fraction of reads compared to the total number of reads that are mapping to the genes. Okay. And this is something that we will discuss next. For the second question here, right, do genes B and C show lower expression in sample 3 compared to sample 1, you have to be a bit careful, right. So, one of the things you probably notice, gene A becomes 0, right. So, it means clearly that gene A's expression has dropped in sample 3. Okay. So, what it means, if you are sequencing up to the same sequencing depth, only B and C will get all the reads, because gene A is not expressed now. Okay. So, only gene B and gene C are expressed. So, all the reads will come from gene B and gene C, but this does not mean that gene B and gene C are showing higher expression, right, because the gene A's expression has dropped. So, we are not getting any read. Gene B and gene C, they are showing higher reads mapped to them. Okay. And this kind of problem, right, this kind of issues will arise, this kind of bias will arise, and this is an inherent bias in the RNA-seq experiment. and you will see a similar kind of problem, right. If gene A is very highly expressed, it will appear that gene B and C, their expression has diminished. It is because the number of reads that you are getting, it remains the same, only gene A will take a much bigger fraction of the total number of reads, but this will reduce the fraction of reads for B and C, and they will appear as down regulated or showing lower expression. Okay. So, this is something that we again have to be careful about, and this is something we have to deal with when we are analyzing the data. So, the technical term that we use is called data normalization. Okay. This is a process that actually tries to minimize these technical errors, and here is the pipeline again for you. So, we have talked about all these steps before: quality control, mapping, assembly and quantification, and this is where we are now, normalization. Okay. And this is the final step before we actually go into the data analysis or actually answering the biological question that we are interested in. Okay. So, data normalization, the goal is to minimize technical variation without affecting biological variation, and we have to do something called sample specific normalization or within sample normalization, right. So, we have talked about some of these bias correction methods. These are actually within sample normalization, and then you also have between sample normalization within a data set. Okay. So, if you are working with multiple samples, as in the example that you have taken, right, we want to compare across samples, then in that case we would have to do between sample normalization, but these are part of a single data set, and sometimes we are working with multiple data sets, and in that case we would have to do something called a batch correction. Okay. So, again this variation from one set of experiments to another we can see, and we have to correct for those. So, for within sample normalization, we have talked about bias correction methods, for example, positional bias, sequence specific bias and GC content bias. We have also mentioned gene length bias as well as sequencing depth. Okay. So, we will discuss this in much more detail, the data normalization method, in the next few classes. Here are the references for this class, and to finally conclude, we have talked about these different types of biases in the data, and these can generate technical variations in the data which we need to account for. We have talked about sample specific biases. These include positional bias, sequence specific bias and GC content bias, and we have discussed about this. And we have also mentioned different bias correction methods. These actually can account for these different biases, right. So, we get improved transcript abundance estimates, and we have talked about some of these tools that we can use for this process, and we have introduced the term called normalization, and this actually this is a method that is aimed towards minimizing technical variation, and we have again discussed this within sample and between sample normalization, what is the need, and we have also discussed about batch correction. So, in the next class we will talk about these different types of normalization in much more detail. Thank you.